Okay. Good afternoon, everyone. Welcome to the IFAS AI and Data Science Seminar Series organized by the IFAS Faculty AI Working Group. My name is Charlie Lee. I'm a professor at the Agricultural Biological Engineering Department. I also serve as the AI Administrative Coordinator at IFAS and a member of the Seminar Planning Committee. The seminar series is part of the effort to build a robust scholarly community on AI and data science at IFAS. It provides a platform for both internal and external speakers to share their research in AI and data science for agriculture, natural resources, and human systems. We welcome your suggestions on future speakers. So today, it is my great pleasure to introduce two of our internal speakers, Dr. Liu from Horticultural Science Department, and Dr. Nikos Tziolas from Water, Ecological Systems and Sciences. So each speaker will present their work in 20 to 25 minutes and leave about 5 minutes for questions at the end of their talk. So without further ado, let's get started. Let me first introduce our first speaker, Dr. Liu. Dr. Liu is an assistant professor in horticultural sciences. He did his graduate study in both Peking University and Yale University, and his postdoctoral research in Stanford University on plant meristem and leaf development. He joined UF in 2018, and his lab focuses on understanding the mechanisms that plants integrate developmental pathways and abiotic stress pathways in model and horticulture plants. His lab is also characterizing the genes and gene regulatory networks that are involved in post-harvest senescence and deterioration using quantitative genomics and gene editing and machine learning approaches. Today, he will present AI and machine learning to reduce post-harvest losses. Tie, the floor is yours. Please share your screen. Okay, this looks good. So Tia, you are muted. Can you unmute yourself?
All right. Okay. Sorry about that.
Good. Okay.
Well, thanks Charlie and Damien invited me to give a seminar on this topic of AI and machine learning. So I want to talk about the work in my lab, basically study post-harvest biology, but we also use machine learning tools to understanding those post-harvest questions and then develop a new technology to reduce food loss. Just a kind of brief background why this is matters, and I think post-harvest biology is really a directly related food waste and loss effect. So as a matter of fact, almost 1/3 of the food never been eaten in not just locally but globally. And food waste is also a huge problem, such as environment problem, but also can cause a lot of the soil conditions or environment and temperature changes. So it's a huge problem in current days and also could be in the future. And vegetable and fruit decay is one of the major why this is a cause of food waste and loss. And this is probably you can observe in the daily basis in your kitchens, different kind of vegetable and fruit are decayed in different ways. So this you're probably surprised. Some of the vegetables are, for instance, have fungus infections. Some is vegetables such as the tubers have sproutings, and a lot of vegetables also have developed this disorders when stored in the lower temperature conditions. This is so diverse. It is also making this very complicated or difficult to study. But what is the really nature? Why does this happened? And that I think is a major um, uh, reason is because those are horticultural crops are fresh produced. They are still alive. They're breathe and they release heat, and they can lose the moistures, and those can also get sick. So because of the nature of those problem, and it really intriguing questions to understand why and what caused those decay and senescence. So how are we going to characterize those post-harvest problem? As you probably see in previous slides, they're so diverse. But in general, we can I would like to classify into three major changes, such as the morphological change. Those are usually involving epidermal tissue change, color, firmness, or weight. The second would be physiological or biochemical change. Those is usually involving water loss, respiration rate change, chlorophyll content change, or even hormone or nutritional imbalance among those tissues. In addition to that, they also have genetic variations. We know that some vegetable and cultivar maybe have a longer shelf life, as more resistant to decay process. And that's could be involved in certain, for instance, gene expression difference, proteins, metabolites, or pathway, or even epigenetic involving this difference. So really to answer those questions, there's a lot of imaging or a lot of detection technology has been developed in terms of visualize those changes. For instance, so we see some of the scanning microscopy image, you can see the cellular level change. Others imaging like hyperspectral imaging were able to detecting surface or external changes. Imaging technologies such as X-ray or CT scanning were able to detect internal, such as those decay process. Those are usually the non-constructive technology. Basically, they are not gonna break down the tissues, and they're still able to visualize those changes. In addition to that, they also have technology where it can visualize those compound or elements changes. For instance, multi or topological imagings. Those are usually the constructive imaging technologies. So both imagings are very have their advantages in terms of understanding those or detecting this decay process. So that's what my lab is to instead of developing a single technology to visualize this change, but we're actually focused on answer different questions of post-harvest biology questions. Those questions could be involving, for instance, broccoli yellowing process, or potato sprouting process, or fruit ripening process. And the way the work is we want to understanding what actually genetic control this process. So this is really through understanding the genetic variations among those decayed vegetable and fruit versus different cultivars. In addition to that, we're also developing different imaging technology. This is usually characterizing morphological or physiological biochemical changes. And then the really central question is how are we going to correlate everything into a meaningful relationship with, among those factors, such as what's the genetic contribution to this, and what is actually those morphological and so to answer those questions, we really answer each of those post-harvest problem, and by design a way to correlate genetic variations versus imaging-based tools. So that's I'm going to give you a couple example basically to answer those questions. One of the examples to study broccoli yellowing process and avocado chilling injury, as well as the passion fruit ripenings. And so that's this work is basically heavily collaborated with Alina Zer's lab, and most of the work in terms of algorithm design are from Shaoli graduate students in Dr. Zer's lab. The first short stories want to talk about what is the genetic difference and how we're going to detect it, broccoli yellowing. And we know the broccoli is plants where yellow if you're leaving it counters or not be refrigerated. However, when you buy the broccoli from the store, usually what you right away is to keep it fresh to refrigerate it. So in the lower temperatures, really can slow down the broccoli decay or senescence process. In addition to that, there's industry actually use different compounds such as ethylene inhibitors, 1-MCP to treat the plants and delay the senescence. Others technologies such as controlled atmospheres can slow down the senescence process. So our first question is, can we use any tools to identify any protein or genes are involved in this? What is actually those proteins? To answer that, we perform RNA sequencing and proteomics. So this is for the people who are not in the biology background. This is a basic technology. You can identify different genes and proteins are involved in a particular process. So for long story short, basically we identify different compounds, different genes involved in this, and we're particularly focused on interesting pathways. One of the pathways is glucosylase pathway, because this pathway are highly differentiated among those conditions. And we think this maybe glucosylase can be used as a monitor or marker pathway to visualize those differences. And it's important to identify those protein genes, but then still we want to understand if there's technology we're able to detect those or visualize those decay processes. For instance, you can take a picture of the plants and you're able to know the freshness of the vegetable and fruit. So that's really, it's important to develop an easy access tools to visualize those changes. So you can imagine this as a use facial recognition system to know each different persons. And you can think about, look at those compound or protein or genes as individual elements that are able to detect use imaging this tools. Then our goal is, can we design or develop a new imaging technology to visualize this? So we look at different imaging, as I presented earlier. So one of the most promising is use hyperspectral imaging to characterizing those decay or senescence process. And now the questions become, can we visualize those or can we correlate those compounds with hyperspectral imagings? So to answer that questions, basically graduate student Tziolas, you can imagine those as the technique is like you characterizing different compound and a different wavelengths. So this graduate student Tziolas, to characterizing this, she's looking at different senescence stage of the broccoli, and then use hyperspectral imaging to detecting those changes over the time. And it's interesting, we do find there's a wavelength shift when leave the broccoli in the room temperatures, and they find in the certain wavelengths around the 700 nanometers, and there's a difference compared to the refrigerated conditions. This is going to give us an indication, perhaps those wavelengths were able to see the difference. And furthermore, we did analyze the glucosylase, and this is also interesting. When we did glucosylase concentration measurement, and we find those plants when storage at room temperatures have lineage increase over the time. And however, those increases are not being detected in the refrigerated conditions. So that indicating those glucosylase can be perhaps can be used as a fresh indicator to detect those senescence process. So kind of put this long story in short, we're just we use imaging technology to detect this broccoli senescence process, and some of the compound potentially can be used as monitors to represent those senescence process. So then I'm going to switch gears, talk another two short stories, which is the detecting another two compounds, another two commodities, such as the tropical fruit, avocado and passion fruit. Avocado is also a very, I think, popular fruit. A lot of people considers avocado as a healthy vegetable or fruit. So this is really important. However, there's avocado also have very significant chilling disorder. So in other words, when plants are, when the fruit is storage in a room, cold temperature in at certain temperatures or a certain period of the time, they will display this decay process, either brownings or tissue decay. And so this is a really cost of food waste and loss. We know that those chilling disorder for avocados are caused by different aspects. For instance, their production areas or maturity ripening stages, all those factors can contribute to avocado injuries. In addition to that, the cultivars also a big roles in terms of determine those chilling injury process. So now the questions, can we use imaging to detect those decay process? So we will also think about use hyperspectral imaging as a way to detect those decay process. So the graduate student, Xiaolei, again, she did several round of analysis. So you can see the round one is basically she treated plants at different temperature conditions. They leave the plants in the 25 degree, which is higher than room temperatures, and then the 4 degree, as well as in -20s. And the reason she's doing this is because they create an extreme condition or temperature condition, and then detect what is actually fruit changes over the time. So then they can use this to train the machine or train their model to understanding what is the two extreme conditions, and then developing algorithm understanding in the condition in the between those process. So interestingly, there's some of the algorithm or she uses actually were able to visualize those changes. One of the approach she used is IFUMI approach that developed by Zer's lab. In addition to that, they also apply those approach to understanding the plant when fruit treated in different temperatures. So this is kind of promising. We're able to visualize those change under different or under 3 different extreme conditions. To validate those results, she did a second experiment, which is use hyperspectral imaging. At the same time, she peeled the plants and then marrying those injury areas. So then we can see there if there's a correlation between the actual injury conditions versus the imaging conditions. So again, she were able to detect those changes and have correlation with those imaging process. So this is kind of promising. The third experiment she also did is to correlate with the firmness measurements. So in other words, she measured the fruit under different treatment in terms of the firmness, and then correlate the firmness changes with the imaging technology. And from this analysis, she also were able to correlate those changes. So use the IFUMI approaches that she were able to detect those avocado under the chilling conditions. So this is also quite promising, and we also want to apply this technology in other vegetable and fruit under the chilling conditions. So kind of summarize, basically, use those chilling algorithm development, so we're able to detect those chilling injuries in the avocado under the cold conditions. So in addition to that, we also know that cultivars also play very important role in terms of the chilling injury in avocados. To better understand the mechanism, we know that under the chilling conditions, a plant basically is a membranes, which is the lipids, play very important roles to controlling this process. So to further understanding this, we apply basically lipidomic analysis. This is the way we identify all the lipids over the time, and also in the plants, either with normal conditions or under the chilling injury conditions. So use those lipidomic profilings, we're able to characterizing what the lipids are different among those 2 conditions as well as different cultivars. For a long story short, we do identify different lipids in response to chilling injuries. In addition to that, there's several important lipids. I just want to highlight one of the lipids actually is the brassinosteroids. So this is kind of interesting. Those lipids were able to use as a potentially marker to detect those injury conditions. So for now, we're basically designing tools or imaging analysis to correlate those brassinosteroids concentration with the used imaging technologies. So last part of the story, I want to talk about how we're going to investigate passion fruit ripening. So this is one of the ripening is a big question for a certain fruit or certain fruit type, because it's really hard to detect when they are ripened, especially before harvest. Those is a challenge to understand for farmers when to harvest the plants. Passion fruit is a very important crop plants, especially tropical crop plants. This is in a certain stage, it's really difficult to tell when the actual plants are ready to be harvested or when they are ready to be eaten. So to understand that questions, we basically need to know like what actually factors to contribute those ripening process. So this is an example by looking at different cultivar of the passion fruit. Those is when you cut it open, there's several tissues and several layers are involving those ripening process. And to answer those questions, we want to know what is actually factors are contribute those ripening process. To understand this, we're performing this is metabolomics and proteomics to understanding what the actual compounds or proteins are involved in ripening process. In addition to that, we also apply this approach in 3 different passion fruit cultivars: passion fruit Tziolas, passion fruit Floricarpa, and passion fruit Lingloris. To keep in mind the reason to select those cultivars, because they obviously have different shelf life. In other words, those fruit have a different decay or ripening process. So we use those approach where we're able to identify different compounds or different protein may be directly involved in ripening process. In addition to that, those compounds could also involve in a post-harvest shelf life. So just kind of, again, for a long stories, we're select a certain pathway or compound to study for further analysis. One of the key pathway we want to highlight is ethylene biosynthesis pathway. This is you can see there each different compound highlighted different colors represent different passion fruit cultivars. For instance, the purple one is the purple passion fruit, the orange one is orange passion fruit. And obviously those ethylene precursors, the concentration are accumulated at different cultivars in addition to their ripening stages. So indicating those ethylene pathway may have a different roles in those cultivar ripening process. In addition to that, we also to looking at the proteomic profilings, there's one of the key pathway involving significant difference, which is the cell wall process. We also think this is quite interesting to investigate further. To answer these questions, we'll measure those ethylene concentrations and the different time points in the passion fruit tissues. And then we see there, interestingly, there's passion fruit concentration or significant decrease in the first 3 time points, and later on they're bounced up again. So this is kind of interesting to see why this ethylene productions are changed over the times. In addition, we also measure the respiration rate to see how this correlate with ethylene productions. The second we want to look at those, how does cell wall component change? This is also interesting phenomenon where we look at the tissues in the passion fruit. Those different tissue layers also change over the time. So by collaborating with our faculty Catalings, and he actually measured a different cell wall component in those passion fruit layers, and they find certain compounds or certain component the cell walls are decreased. Some of those are actually increased. So this is going to indicating during those ripening process, those compound could be really involved in ripening and used as a marker to indicating the ripening process. So for that, this is interesting. We further want to see, is there any imaging we can detect those processes? This is actually use a CT scanning imaging to look at the passion fruit before ripened and also after ripened. This is more wrinkled, severe ripened process. And use a CT scan imaging, we're able to actually see dramatic differences. This is a CT scanning of the passion fruit before ripened, and the same as we're looking at the ripened passion fruit. You can see there's obviously very different. One of the tissue is several of those cell wall layers are very different among those tissues. In addition to that, this graduate student Xiaolei also did hyperspectral imaging, detect those passion fruit ripening stages. And the blue indicating the healthy tissues, the yellow indicating the decay tissues. So using this approach, they were able to detect those changes over the time. So this also can obvious from this hyperspectrum algorithm end member calculation can see those shift in certain cultivars versus some other cultivars. This is indicating those hyperspectral imaging were able to detect some of the cultivar at certain storage conditions. Now we kind of correlate all this interesting data together to see how we actually can manipulate those changes and design a new way to monitor those differences. So I hope I convinced you those are basically my lab is focused on each different post-harvest questions, and we want to use both omic analysis, also machine learning tools to not just understanding the genetic difference, but also use tools to visualize those changes. And those would be a really important or powerful tool to understanding different post-harvest questions. So in addition to the questions I mentioned earlier, but we also use similar to the understanding, for instance, such as potato sproutings or strawberry ripenings or decay process, and also other post-harvest questions. I think for that, I just want to thanks for most of the algorithm design and calculations or hyperspectral imaging studies by graduate student Xiaolei Guo in Alina Zer's lab. And also the undergrad student Braden Heimberg did some of the passion fruit works. Of course, our collaborator Alina Zer's really a huge support on this project, and she has a lot of ideas to help. And Kathleen did a cell analysis. Also, the Maoli from Danforth Centers did some of the X-ray imagings. For that, I thank you for your attention, and I'm happy to answer any questions.
Okay, great. Thank you, Tie, for your excellent presentation. Any questions from the audience? You can just unmute yourself and ask the question. You can also send your question to the chat box. Either way is fine.
I just want to thanks for everyone audience here. And if you have questions on the biology side, I'll be happy to answer and probably can't answer the question, but some of the algorithm design or mathematic modeling, that's from Alina Zer's lab. So she will be the expert to understanding those why certain methods has been used or applied to this approaches.
So Tie, I have maybe a comment or question. So for this hyperspectral imaging visualization of a vegetable crop senescence, so I'm just curious, what's the kind of goal? Is the goal to let this use this method for plant breeders, researchers, or for vegetable packers or growers? What's your vision for the next step?
Yeah, I think this is exactly the question we want to answer for to help growers from a research aspect. It's important understanding mechanisms, and also from plant breeders to aspect to identify new cultivars or germplasms that have more better quality or more advanced quality. But for as the growers aspect, they want to know something. They want to have something easy access. They want something quick. So that's the ideas behind this project. We're in the long run, we really want to develop some easy access tools or handheld devices that were able to detect or tracking this process in a faster way or in the more user-friendly ways. I think that's our long-term goal.
Okay, great. So Kathleen, you have a question? Go ahead.
Yeah, nice presentation, T. I was interested if you think that hyperspectral imaging could also be used for passion fruit, since in contrast to the other produce that you showed, passion fruit actually should look kind of wrinkly and not, it doesn't look good from the outside when it's actually ripe and ready to eat. So do you think it's still applicable, the imaging?
What we like to do is actually not for the ripened tissues. We want to develop imaging tools to detect unripened or the tissue in the transition from ripening tissue to ripened tissue. This is kind of stage is a really subtle change in the morphological level. However, must have a lot of physiological and biochemical change among those. So, but those are really hard to visualize, and that's why the goal we want to see, for instance, cell wall changes among those tissues. Is there any way we can use imaging to detect this? For instance, there's correlation among cell wall component change versus imaging tools. I think that's exactly what we want to develop currently. Yeah.
Thanks for clarifying.
Okay, so we have one question from the chat box. Was any time series phenotypes extracted by models like LSTM, GRU used in tandem with CNN for predicting the qualitative traits of fruits, vegetables?
Well, that's the model question, so I really can't answer that question, or didn't know how to answer questions, but I'll be happy.
Okay, so your collaborator is not in the audience?
Yeah, I'd be happy to forward your question to
Okay.
Dr. Bear.
Okay, all right, that's okay. Yes. So because of interest of time, let's move on to our second speaker. Thank you, Tie. So let me briefly introduce our second speaker. So our second speaker is Dr. Nikos Tziolas. Hold a second, let me get his bio.
In the meantime, I can share my screen.
Is assistant professor. Yeah, sorry. Yeah, just found your bio here. So he's an assistant professor right now in the Southwest Florida REC Research Education Center. He received his PhD in soil monitoring with artificial intelligence from Aristotle University of Thessaloniki University in Greece. Yeah, sorry. Yeah. I may not pronounce this name correctly, this university in Greece in 2022. He has extensive experience in using remote sensing and AI techniques in land and soil degradation, compliance checks, and the monitoring of sustainable development goals. His current research involves the assessment of potential of multispectral and hyperspectral sensor systems developed on Earth observation platforms to facilitate the precision mapping and monitoring of soils. So today he will be talking about from soil mapping to informed decision-making for ecological health, a envisioned target and the role of AI. So Nikos, yeah, the floor is yours.
Thank you, Charlie. I can understand all the Greeks are really difficult for you, so what's my presentation? A second. I'm going to share my screen. You see my presentation?
Yeah, you can make it presenters mode. Presentation mode.
I'm already sharing. Can you see my presentation?
Not yet. It's not in presentation mode yet. Now it's okay. Now it's good.
Oh, it's good. So you can see the presentation mode. Okay, yes. So I'm Nikos Tziolas. Sorry, give me a second. Yeah. And I'm going to present very, very shortly how we can move from soil maps to and predictions to informed decision making using, of course, different data and artificial intelligence. So it is changed or not? It seems it is not changed in my screen. Okay. So the content of the presentation today is very, very simple. We are going to present the current challenges and needs, and then we are going to present what we envision as a soil digital twin. And then we are going to provide 2 examples about ICT tools the soil data cube and the soil scanner, and what is the role of AI in order to improve the predictive performance of these tools, and along with some future steps and what we envision and what we have already implemented here in Florida, considering these two tools. And the last but not least, for sure, we are going to provide the team of the recently established Soil and Science and Artificial Intelligence Lab. So just to start, why soil matters, why soil is so important for us. We are at a crossroad in history in our attempt to preserve global soil resources. So the overexploitation causing the Dust Bowl is still within living memory. Soil erosions might increase up to 60% in the next 30 years. And the recent studies here in the United States indicate that the USA may lose around 2 petagrams of soil organic carbon under climate change the next years. So in this direction, in order to safeguard natural capital of soils in this continuously volatile landscape of reduced resources and environmental changes, and in order to pave the way for the development of evidence-based conservation recommendations and policies and sustainable services for relevant economic operators, it is essential to enhance, to improve the capacities for soil health monitoring by adopting multidimensional and integrated approaches. Just to mention in this direction that credit rating schemes are moving fast in Europe and the US. Biden-Harris administrations announced recently new investments to improve monitoring, reporting, and verification of greenhouse gases, including soil organic carbon. So this is a high priority in different agendas. So what is the challenge that we face in order to overcome the current problems? The challenge of scaling. The current products are with a gross spatial resolution of around 250 meters and are based on legacy data that are not up to date. So we don't have accurate information about the current situation on the fields. On the other hand, we have the challenge of cost reduction. Current cost of measuring soil carbon needs should be reduced by around 90% if we would like to talk about a valuable business model, considering carbon farming, for instance. And last, again, but not least, the problem with time consuming. In order to get back a soil laboratory analysis, maybe it's required more than 10 days to get the results to the farmers, to the end user, to the policymaker, in order to support informed decision making. So based on all this, in the recently established Soil and Artificial Intelligence Laboratory here at the Southwest Florida Research and Education Center, we envision to create a digital infrastructure representing the physical environment on the one hand into a big data environment. So we will like to have the infrastructure to collect, analyze, integrate enormous amounts of soil and soil-related data to support informed decision making. So the team here is going to develop ways to incorporate state-of-the-art and novel techniques from such as soil spectroscopy, Earth observation from satellites or UAVs, DNA genomics or whatever. And moreover, we would like to explore the combination of conventional and also emerging measurements from daily to annual data, from quantitative to qualitative information into this digital artificial intelligence environment. So in our mind, the soil digital twin will be based on the substantial developments of in AI methods, such as deep learning regressors in order to perform predictive analytics, edge computing in order to enable processing at greater speeds and higher volume at real time, federated learning to train the data independently without the need to sharing the information among the different users. So this is the vision of the soil digital twin. In order to support this vision, we are going to develop, and we have already developed some tools. First of all, we have the soil data cube. That is a pre-operational soil monitoring system able to provide estimations of various soil properties using Earth observation data, large soil data archives in order to train the models, and of course, AI techniques. And the goal here is to improve both the spatial resolution of the products. As you can see in the GIF, we have the current situation, and on the other point is the enhanced situation, so we can discriminate the difference within the same field compared to the current state of the art. But we would like also to enhance the accuracy of the models. So we are not going to develop everything from the scratch. We have already performed some work in Europe, and we are going to apply this technology here. So in a previous project called World Soils, a project funded by the European Space Agency, we have developed the complete pipeline in order to generate the soil spatial products. So different technologies from cloud computing, parallelization, Dockerized functions, different pipelines in order to address completely different the bare soil predictions and the predictions of soil under vegetated areas. So we have also soils under forests. So this is a completely different approach. I'm not going to explain everything in this system, but I just want to mention that different components are a piece of a bigger puzzle in order to get the final result. So remote sensing, Earth observation by satellites give us tremendous opportunities in order to have a regular monitoring. But we have only 2 small windows during the crop phenological stage at harvesting period and during the emergence in order to recognize the bare soil and develop a direct relationship between the independent variables, that is the data from the multispectral, from the remote sensing, and the dependent variables, that is the soil properties. But definitely we are not living in an ideal world. We can see the bare soil, but the severe disturbance effects on soil reflectance from remote sensing is introduced by partially covered vegetation, both the green and dry vegetation. And this is very, we can see this phenomenon very often in the agricultural areas.
Hey, Nikos, we lost your audio. We cannot hear you. You check your audio device. We still cannot hear. Your audio was okay at the beginning. Can you unplug and plug in your maybe a microphone? You cannot hear. Maybe you log off from Zoom and log in again. Yeah, sorry about this technical difficulty. Yeah, so yeah, I guess you are still there. If the audience, does anyone have any questions to our first speaker? I think feel free to ask.
I think maybe you can give a brief introduction of this seminar series so we help audience maybe can attend further in the future.
Sure. I saw Nikos is back, but we still cannot hear you, Nikos. Yeah, I mean, so Tia, you mentioned about this seminar series. I think at this point, we are forming the seminar planning committee. I think Tia is also one of the seminar planning committee members. And we welcome your suggestions on future speakers. So in particular, external speakers. We currently are developing the seminar series for external speakers in the next semester and the following year. So we will, publicize our committee members' contact information on the website, and you can contact any of the committee members later. So we hope to get that done maybe in 1 or 2 weeks. Yeah, again, if you have any questions to the first presentation about post-harvest study, feel free to ask.
Can you hear me now?
Yeah, Nikos, now we can hear you. That's great.
Yeah, I changed my computer, obviously. I don't know what happened with the microphone, probably. We have a power outage sometimes here. So I'm going to continue.
That's okay. I think you can resume.
Yes, give me a second to open my presentation again. Can you see the presentation mode now?
Yes.
Okay.
It is the first slide. If you can go to the slide you just left.
I think I was somewhere here when you mentioned that you cannot hear me.
Yes, after this slide, I think, probably.
Okay, I have to change manually the slides. So as I mentioned, we have this impact of the disturbance effects on the soil reflectance from the satellites, and we can see in the plot that the ideal scenario is the red line, but we have different recordings at real field conditions as recorded in the great light in the light gray lines. So in order to overcome this problem, we introduced a data mining technique when we analyze the time series of the remote sensing data from Sentinel-2, the multispectral data, in order to discriminate and then mask out all the other crop types and land uses, and in order to keep only the bare soil pixels. This enables in order to do this, we apply a generic methodological histochrome separation thresholding. We analyze, as I mentioned, all the time series, and finally we create a bare soil composite. What is the bare soil composite? Is a temporal composite that can be used instead of a single-used multispectral image to account for the frequency of vegetation cover of soils and give us an opportunity to get a broader spatial coverage of bare soils. So as we can see, trying different months, we can have different results. For more area that we can observe in at real field conditions. So these are some examples. We can generate a cloud-free composite. It's really important for agricultural applications. We can see also at very high spatial resolution the bare soil frequency in different months, in March, in June, in November. Obviously, we have a different percentage because we have the different crop phenological stages. But also we generate, as I mentioned before, the bare soil reflectance composite.
And this is really important because we utilize the bare soil reflectance composites, in order to generate later on soil spatial products. So we utilize these multispectral composites as input, and we leverage the techniques from one-dimensional convolutional neural networks algorithms from the family of deep learning in order to estimate the soil organic carbon content in this case, but in general it can be applied in different soil properties. From the one-dimensional spectral signature, we have this as our input, and we apply different pre-treatment techniques. In order to, such as conversion to absorbance or application of standard normal variate, in order to enhance some absorption bands or perform a scatter correction that can be seen from the model as different channels. So doing this, we would like to exploit the complementary information of the various channels by maximizing the spectral space, and this allows us to get a better predictive performance for the soil property at the end. Based on these predictions, we provide always also the uncertainty layer in order to guide the end user about the uncertainty level of the predictions. So instead of just present some static maps, we can have a closer look from this interactive map. It is from the country of Lithuania, an example in Europe. We generate various soil property maps from soil organic carbon content, but also more advanced products such as soil organic carbon sequestration after applying process-based model techniques, and we validate this approach. So based on this, the end user has the opportunity to go at specific fields, see the results, see the predictions, and they can download the data for further analysis or for informed decision making. Definitely, this is a short demo, but you can play around with the QR code, and you can visit the site in order to explore the viewer, the carbon monitoring tool. So just to summarize, we saw the bare soil composites that allow us to have a temporal generation of different products at annual, monthly, or on demand according to the needs of the user. We saw examples how we can eliminate the effects of ambient factors applying data mining techniques, and also we saw how the bare soil reflectance composite can support the broader spatial coverage of the agricultural areas. In terms of AI, convolutional neural networks, we develop our own algorithms in order to enhance the predictive performance compared to the current state of the art. We achieve a better predictive performance around 80% compared to random forests, for instance, or SVM. But also the multi-input convolutional neural networks that we present give us an opportunity to enhance the predictive performance considering the single reflectance convolutional neural networks. Last but not least, of course, the cloud computing performance give us an opportunity to analyze big data, big Earth observation data at national and continental scale. We can have an automated generation of different spatial products, as I mentioned, at different levels, at annual level, at the monthly or on demand. And for sure, we can reproduce this technology at the different regions. And based on this, the first month that I'm here, I have just 5 months here in Florida. We have already developed a data cube oriented for the Florida state. We have already analyzed the Landsat and the Sentinel-2 archive to satellites providing us multispectral data in order to generate this bare soil reflectance composite that can allow us to generate spatial products at very high resolution. So we are going to increase the spatial resolution from 100 feet to 32 feet around in order to support informed decision making at the field level. So this is the first stage. We are going to utilize the Kellogg's soil spectral library from the USDA in order to develop the models and predict soil organic carbon, pH, clay, other parameters that can support our vision to present a soil health radar for each field in order to give an opportunity to the farmer, to the grower, whatever, monitor at monthly basis the field. For sure, we are familiar about the challenges that are existing in in the environment of Florida, and we are going to overcome this by introducing other techniques, but we are still in the phase of development. So this was one example about soil data cube. Another example is the soil scanner. Overall, a host of novel and potentially low-cost spectral radiometers are rapidly maturing and can become a viable alternative to costlier traditional solutions, the chemical laboratory analysis that I mentioned at the beginning. So we develop a low-cost sensing tool, leveraging again the soil spectroscopy, AI algorithms, and of course processing on the cloud to support stakeholders for in situ surveys and soil testing advisory services at the field scale and at real time. So we develop advanced data analytics for processing and validate both data and metadata. We develop AI algorithms in order to transform the spectral signatures into meaningful information. We will see later on what I mean by this. And also the user-friendly interface to give an opportunity to non-experienced user to use this technology. So I have to mention that nothing is plug and play. So we start thinking that maybe this will be an easy task. We get a sensor from off the shelf, a small, low-cost spectral radiometer able to capture the spectrum in the SWIR region. And we think that we can use directly this in order to capture the data, translate this data in soil properties, and give the result back to the end user. But we realized that the distance between soil sample and spectral radiometer must be increased due to the small viewing of the apparatus. So we refresh our memory with Beer-Lamberter law back to the basic principles of physics, and we start to develop a custom 3D printing solution. So we use 3D printing in order to modify the initial version of the sensor and to conclude to, let's say, a sensor that fits in our needs and can generate accurate results. So overall, in order to ensure that the quality of on-site in field collected data is not compromised, we develop an AI-driven outlier detection mechanism. So sometimes we utilize AI just for regression analysis, but we can have AI as an outlier detection component. So we develop a small data set from spectral signatures from everyday objects. We classify an AI classifier, an ensemble classifier, and we get a very good accuracy in order to remove the outliers. This is really important in order to give accurate predictions later on. So we use also AI this, but for sure, we use also the AI in order to give us an opportunity to leverage the existing information that is contained in big soil spectral libraries. It's not necessary again and again develop the calibration data sets. There are existing soil spectral libraries. So we have in our mind, let's leverage this information in order to calibrate our new models. So in order to do this, we saw before that there are several ambient factors that influence the spectral signature of the soils when we perform the analysis at the real field conditions. So we conceptualize a signal denoising algorithm. We use again convolutional neural networks in order to minimize the spectral distance between the in situ and the laboratory reference spectral values that we have in the existing soil data archives. So we achieve a very good result, and then in order to exploit these data sets, we develop, no, we don't develop, we apply AI regression analysis with common state-of-the-art regressors such as Cubist, Random Forest, in order to predict calcium carbonate, clay, and organic carbon. And as we can see in the scatter plots at the end, Of course, we have an accuracy loss was in the modeling of in situ spectra compared to the pre-treated laboratory analysis, but the results are quite good enough at this case. Otherwise, we cannot predict a direct the soil properties using the spectral signals that we capture at the field. So we have already developed, as I mentioned, the interface. We give the different sensors to the farmers and to the growers in Europe. We have in 2 different countries more than 2.5K observations. So the user goes directly to the field, scan the field, and get back the prediction. As we can see here, we have an example in Lithuania. We always ask for them at this development phase to capture the reflectance data for calibration, but the algorithm automatically translate this into, let's say, a laboratory spectra in order to perform the analysis. We always ask for the user to provide us also a photo from the field in order to be sure that they are measuring bare soil, the bare soil, and we deliver, as you can see, the prediction directly to their phone. So the soil organic carbon content here is around 5% in both fields that are very close. So in the next steps, our laboratory initiates, together with the University of Sao Paulo, the development of a GLOSPEX. It's a global online soil analysis system. Till now, we have collected data from more than 90 countries in all over the world, soil spectral data. We have also collaborators from the University of Florida. Sabine Grunwald is going to participate also in this effort. And the overarching objective here is to develop a system that, where the user can upload the spectra, and the system choose the algorithm and return back the prediction by email or by text. In this case, we are going to leverage multi-input convolutional neural networks, but we are planning to develop a localized adaptive correction mechanism in order to enhance the predictive performance, because we are talking about a global data set. Another step that we are going to implement the next months is the application of a federated AI framework for soil analysis. So recently we informed by the GFSI that they are going to fund our idea about the Flora project, where we are going to create a collaborative example of AI regression algorithms, allowing different participants, different organizations, in our case, different soil laboratories, to participate in a modeling effort, creating personalized models without directly sharing their data. So the user with federated learning keep their data, but they are sharing the models and the weights of the models. To conclude, I truly believe that AI can make the difference, but always the people is the driving force that make the difference. So the first 5 months, we focus on the development, on the recruiting of our lab. We have already our first data scientist and our PhD student, but we have also the privilege to collaborate with a spectral lab from the Aristotle University of Thessaloniki, one of the most prestigious laboratories in Europe in the domain of soil analysis, artificial intelligence, and remote sensing, with more than 25 researchers and more than 30-40 ongoing projects in this domain now. So we have this privilege to collaborate and extend our network at international level. So that's all from my side. I'm really sorry for this misconnection. I hope that I'm still on time. Yes, I think.
Yeah, we are about 4 minutes behind. So we certainly understand the technical difficulty. Sometimes it happens. So let's just accommodate one question, and then we can join our seminar. So we have one question from the audience chatting box. Vectan asked the question, on scatter plots, I did not notice the soil depth. What is the soil depth? Your approach can provide reliable results. Nikos, can you answer that question? Can you unmute yourself?
Yeah, sorry. Yeah, I have a problem with with the sound this morning. Are you asking about the soil depth? It's correct?
Yeah.
It's
In
topsoil.
the chat box, yeah, you can
It's
see the question.
it's
Soil
topsoil.
depth.
It was a topsoil measurement. As I mentioned, we share 30 scanners in different growers and advisors, and we ask for them to scan the topsoil layer. So the results that you saw are the topsoil from 0 to 30 meters. This is the reason that sometimes we ask also for them to provide us a photo in order to be sure that they have measured the soil layer.
Okay, all right. Because we are kind of late, 6 minutes over time. So if you have more questions, I suggest you send an email to our speakers. So again, I want to thank our two speakers for their excellent presentation. I want to thank everybody for your interest and time attending our seminar. So again, if you have questions, please send an email, reach out to the speakers. Thank you very much. I look forward to meeting with you all in our next seminar. Bye, have a good day.
Thank you.